First of all, thank you, Royal Society for admitting me as a new fellow. I'm really honored and also humbled. I was born in China and did my bachelor and master degree in China, then moved to America, did my PhD in University of Pennsylvania in virology. Then I came to live in New Zealand and working on human viruses for the last 27 years. So today my talk is about the science and impact of SHIVERS. Apology for the wrong slide sets. So first of all, the influenza virus is an RNA enveloped virus, as you can see over there. And it has 4 types, A, B, C, D, and the influenza A is a real troublemaker because its natural reservoir is waterfowl. Sometimes it can spill over to human. If the virus is able to transmit in human population easily, then it will cause a pandemic. And we had a pandemic in the past over 200 years. And the most severe one, you probably heard, the 1918 pandemic. And it killed 50 million people. The most recent one is 2009 pandemic. And look into the future, pandemic will happen. It's just, we don't know when it will happen. So the best strategy for us is to prepare for that. So why SHIVERS? Soon after 2009 pandemic, WHO Review Committee concluded the world is ill prepared to respond to a severe influenza pandemic or any similarly global sustained public health emergency. So in 2010, US CDC made a funding opportunity announcement, welcome applications from Southern Hemisphere country with a temperate climate. And it's very competitive. We were pleased that we got the award because New Zealand has a really excellent public health infrastructure. We all know each of us have National Health Index, which is really powerful for data linkage. And also, most of New Zealand are registered with their general practitioners who kept their very good medical records. And the New Zealand population is really well characterized, including itenicity and the social economic status. So we call our project as SHIVERS, and now it's the first iteration of SHIVERS. So what we did is we used Auckland as the study site, and we established a hospital surveillance, and also established Sentinel General Practice surveillance, and another is a human longitudinal cohort. We have a range of objectives, quite broad, from severe disease require hospitalization to moderate disease require GP consultation, and to mild or asymptomatic infection in the community. And the impact of SHIVERS-1 is to inform the policy change in New Zealand and also internationally. And also we have over 40 peer-reviewed publications, and our surveillance become the national surveillance infrastructure. And we received a few research awards, and NBA accepted SHIVERS as one of the 5 impactful case study. And our new knowledge gave us an opportunity to receive over 60 million national and international funding. So I want to change the gear, give you 3 highlights of SHIVERS-1. The first is the impact on the vaccination policy. If you look at this graph, the red one is about hospitalization from by the different age group. And you can see very young and very old had the highest hospitalization rate. If you look at the blue graph, it is showing in the vaccination rates. And the children under 5 years of age has very low vaccination rate. So this information leads our Ministry of Health to change the vaccination policy and the free vaccine for very young children under 5 years who have been hospitalized or have a history of a significant respiratory illness. The second highlight is SHIVERS on pandemic influenza severity assessment. Soon after the 2009 pandemic, the World Health Assembly passed a resolution mandated WHO to establish a framework for PISA. The goal is really to inform the national and the global and the regional risk assessment for pandemic response decisions. So WHO showed the tab SHIVERS, one, because it's a high quality data. So we participated with the WHO lead pilot on pandemic influenza severity assessment and published this guideline for all member states around the world. The third is our SHIVERS series survey we done 2015. So what we did in that year is we did something new, something novel. We tested each sample, not just a sample against the flu virus hemagglutinin surface protein, which is the most abundant protein, but we also tested the antibody against the neuraminidase, which is a less abundant surface protein, only 30% of those. To our big surprise is actually human generate antibody against the NA much more so than the hemagglutinin. So it's really, this is something is mind-boggling to understand in the immune response, because current vaccine strategy is trying to stimulate strain-specific antibody to HA. The problem with that is HA is faster mutating, and you have to have annual vaccine strain selection in order to have a best match for the circulating viruses. Another problem is it ignores the NA, because most of the manufacturers don't have a standardization process. So in reality, every time when you have an annual jab, you have a very low quantity of the NA amount. So it's really the time for game change. We need to think outside the box of the HA. And at the moment in New York, Mount Sinai Medical School, they are conducting clinical trial for the NA. So when we finished the SHIVERS 1, we were very grateful that our Ministry of Health continued to funding hospital SARI and GPII surveillance. And our series survey result helped us to attract funding from US National Institute of Health. We established SHIVERS 2 adult cohort and in Wellington region. So we also call it as a well-Kiwi adult. And then we established SHIVERS 3, the well-Kiwi infant cohort, and also SHIVERS 4, well-Kiwi household cohort. And really the main objectives, the primary objective of these three cohorts is really trying to understand immune imprinting, particularly about how and when and why when you encounter the flu for the first time, or maybe previously in your lifetime, and how that immune response will be impacted for you to respond to the second or the third flu exposure. And our really important understanding is to determine the correlates of protection. And I won't have time to go through all the detail with you, but one of the study we did is about the immune response and published in the it's over here in the Nature Immunology paper, and it's really exciting, but I don't have time to go through that. And this week in Virology made a podcast. It's in a very easy, understandable way, so you can access from the YouTube. Yeah, I won't go through all those details over here. So the last thing I want to mention very quickly is the impact of COVID-19 public health social measures on flu, RSV, and SARS-CoV-2. When we had a public health measure like a lockdown and the border closure, actually we eliminated the SARS-CoV-2, but we also eliminated flu and the RSV. And this is basically the red line shows all negative flu viruses. So what we found is New Zealand's platform is really valuable to understand the virus circulation, and this is really something we are able to do for this particular unique island situation. So our publication actually attracted funding from Flu Lab, and which is a philanthropy foundation, and they funded us for Shivers 5 and then for Shivers 6 in the next 4 years. And we're trying to understand the impact of immunity gap on influenza circulation and influenza severity. So I probably will end over here. And this is a few conclusions from our study. And really just want to acknowledge hundreds of people in the past decades and devoted their time, energy, and their expertise for this really successful multi-center, multidisciplinary collaboration, and also to thank my family for their support all those years. Thank you.
Thank you. Well, thank you for that tour de force through influenza. I enjoyed it. Missed a lot of the detail. But one of the things I'd like to ask, if I may, is with recent changings and perhaps gutting of some of the U.S. agencies and the evolution of the flu virus and some of the different strains in that, how does that affect trying to work out the efficacy, if you like, of a universal vaccine?
Yes, this is really a big challenge we face because the virus doesn't recognize the country boundary and it moves around across the border. We really need to have a cross-country, multi-level, lateral kind of a collaboration, and to see, USA withdraw from World Health Organization is really a big blow for all of us working in the public health. However, I think scientists think in very similar ways. Even though with the constraint like that, we actually still managed to select the vaccine strain. For example, in February this year for Northern Hemisphere country, with the CDC still have scientists involved because the world is so much interconnected. And we have to have information from all different countries in order to make a sensible vaccine strain selection. But we are working in a different ways, but still collaboratively, globally for that.
I'm sure you have other questions. Come on.
When you talk about the monitoring, what kind of data do you collect other than just fatalities and vaccine rates?
We collect a whole range of the data sets. Mortality is something everybody wants to know because the minister, once they hear whether there's a pandemic come along, the first question is how many people would die from this infection. So it has such a huge impact. It is the one keep the politicians, keep the modelers, really the first thing they needed to know. But the impact on the hospitalization is another very important aspect. However, that is really just the tip of the iceberg. The things, what's happening in the community is the things gradually lead the progress into the hospital and cause a huge mortality and huge morbidity. Therefore, you have to collect the mild infection data as well. And that is the one allow you to understand exactly what is the burden in the community and how it will eventually lead to the severe impact. And then as a human population, all the interactions around the society, you do have to collect the behavioral, a different setting in the school or even in the nursing home, in many different settings. Only when you have a variety of data, then you will be able to model the complexity of the interaction between the viruses, between the human host, and between the environment.
Well, I have at least 30 more questions, but unfortunately we have to end it here. So thank you again, Sue, for a wonderful talk. And
Thank you.
congratulations.